will introduce our two speakers. Our first speaker today is going to be Dr. Julia Stone. She is a senior research fellow who recently joined MSPS last year. She's an NHMRC emerging leader, and she's a chronobiologist with training in sleep and circadian rhythms research. Her research program seeks to understand and model circadian disruption and its impact on health and well-being. In particular, she works to improve methods for tracking circadian timing in populations with sleep disorders and in developmental contexts such as adolescence. So we are very excited to hear from her today on novel approaches for tracking circadian disruption. Please join me in welcoming Dr. Julia Stone.
Thank you. Thanks so much for having me speak in this session. As Sarah mentioned, I moved to the school last year from Monash, and I've slowly been sort of getting to know people, but there's lots of people I haven't really met or spoken to yet. So I think this is a really nice opportunity for me to kind of talk to you about the stuff I'm interested in and the kind of work that we're going to be doing. So in particular, I'm going to talk about circadian disruption and what it is and how and why we should be measuring it. And then I'm also going to talk about some new emerging approaches that we're playing with to try and sort of track circadian timing out in the wild, if you like. So largely a background talk, but then a bit of a data treat at the end. So our bodies are made up of clocks. We have clocks in all of our cells, all of our different systems. They are responsible for regulating the timing of basically every biological, physiological process in the body that you can think of. And we have a central circadian clock, which is located in the SEN, and its job is to coordinate all of these different clocks. And it is synchronized to our 24-hour solar day, primarily by exposure to light. When our body clock becomes misaligned from the solar day, we have what's called circadian disruption. And that has really broad consequences for our health, for how well we're sleeping, for our well-being and our cognitive performance as well. And really, this type of dysregulation can affect all different levels of the system, and it can occur in different kinds of ways. So we can have misalignment between our environment or our behaviour relative to our internal body clock. So for example, you fly to Paris and you need to live and sleep on Paris time, but your body clock is still on Melbourne time. You've got misalignment there and you will feel jet lagged and you will feel terrible. And then we can also have misalignment within our internal circadian system as well. So between all of our different peripheral clocks, they can kind of get all jumbled up. So for example, you're sleeping on Paris time, your body clock is still stuck in Melbourne, but maybe your liver clocks and your digestive system is somewhere over Singapore time, maybe your cardiovascular system is somewhere else. They're kind of all in disarray. They're a mess. We see really clear examples of circadian disruption when we look at shift workers in particular, because usually they will have to be at work and alert when their body clock is telling them really strongly to be asleep, and then they go home and they have to go to sleep when their body clock tells them to be awake. So we have misalignment between their body clock and what they have to do. We also see examples across different sleep disorders. We've got really extreme cases with non-24-hour sleep-wake phase disorder, which is effectively a failure to entrain to the solar day. So we'll see that their body clock will just kind of keep shifting a little bit later every single day, all the way around the clock. Sometimes their sleep will shift with it, like one of those plots, and sometimes they will do their best to kind of live on our 24-hour day, but their body clocks keep shifting. So there's all kinds of misalignment and desynchrony that comes along with that. And then, of course, if you're experiencing jet lag, you're in a state of circadian disruption as well. And increasingly what we're learning is that circadian disruption is linked to really serious and broad-ranging health consequences that I will show you here. And so that includes things like impaired cognition, mood disorders, cardiovascular disease, we've got hypertension, diabetes, gastrointestinal disorders, immune disorders, increased risk of some cancers. It really affects all systems in the body. And we think that this link is probably bidirectional, like circadian disruption can increase disease severity and incidence, but also many diseases can disrupt circadian rhythms and kind of feed into an endless, horrendous feedback loop. So we need to measure the clock for a lot of different reasons. One is to get a better understanding of the system. We have to measure it in our experimental work to understand the properties of the clock and what happens when it's disrupted in different contexts and how that relates to the regulation of different physiological systems. But there's also a lot of examples where having a measure of the clock has really direct applications for health and safety. So for example, it's highly recommended for circadian sleep disorders that we measure the central clock to help inform your diagnosis and then subsequently the treatment. However, very rarely do we do these kinds of measures clinically, but part of why it's so important is we see these different phenotypes over here, which look really similar if you go by sleep complaints. They'll come in, they have trouble getting to sleep, they have trouble staying asleep, and some of them will be trying to sleep at the wrong biological time and some of them won't. And if you don't measure their biological time, you don't know whether you're treating a circadian disruption with like a light therapy or a chronotherapy, or if you're treating something else like more of an insomnia with a CBTI. Measuring the clock is also super important for our chronotherapies. So one example of that is light therapy. These therapies are phase dependent. So we know from our phase response curves that the biological time that you administer light will determine the direction and also the magnitude of the response that you get. So with light as an example, if you get light exposure before your core body temperature minimum, that will delay your clock, it will push it later. Whereas light after your core body temperature minimum will advance it, it will push the clock earlier. So if you don't measure circadian timing, if you don't know where that person's core body temperature minimum is, you don't know when to apply the therapy and you could end up applying it at the wrong time. And this is likely true across a lot of other chronotherapeutic applications across medicine that are starting to emerge that will be based on their own phase response curves. So there's a lot of evidence coming through now with chronoimmunotherapy for cancer treatment, where basically we have more effective immune responses to the treatments at particular biological times. So you can optimise therapy if you time them according to the biological clocks. And similarly, we're also often really interested in measuring the clock in occupational shift work settings because we have really clear known rhythms in our alertness and our cognitive performance that makes working during the biological night a time of really high accident risk. So if we want to evaluate risk, we want to develop fatigue management plans, we want to create personalised interventions for shift workers and know when to apply those countermeasures for them, all of that benefits from knowing someone's circadian timing and when they're most likely to be at risk. And for all of these cases, I'm going to sound defensive here, but you do need to measure phase. You can't just guess because we see really large individual differences in circadian timing, even when people are on the same schedule. So on the same time zone, they're sleeping at the same time, they've even got the same light input to the clock. We still see really wide variability in where their body clock sits. So example here, hang on, I can do a pointer. The example here is from a group of really healthy, regular sleepers that were either sleeping all at the same time early or all at the same time late. And this is their melatonin onset time, so that's our marker of body clock time. And you can see that even in those really controlled conditions, there's more than a 4-hour range in where their body clock was. And when we look at this in shift workers, this is exacerbated even further. So what I've plotted here is data we collected looking at melatonin timing in shift workers on day shifts and then again when they went over onto night shifts. And what we see is over a 6-hour range in what their body clock time is, even though they worked exactly the same schedule. And we also see huge variability in like how much they will shift and the direction that their body clock goes in. So if you don't know someone's biological time, it's really easy to mistime your chronotherapies, which can make it ineffective or it can actually make it counterproductive as well. So in humans, we can't directly measure the central clock in the SCN. So we will measure marker rhythms that have really well characterised, reliable endogenous rhythms. Usually we will look at melatonin. We also have robust rhythms in cortisol, in core body temperature. And what we're looking for is markers of this grey section, which is our biological night. So we might look at the onset of the melatonin timing, the midpoint, or the nadir in the core body temperature. And these are markers that we have really good protocols for measuring. We can measure them, we should be measuring them lots, but you do need to measure them in really carefully controlled conditions and over a long period of time. So you might need someone in dim lighting for 7 to 24 hours, awake the whole time, sampling a lot of time. My students in the back know about this. They've spent a lot of time in the lab doing this. So it comes with a lot of challenges. It's really expensive in time, in money, in resources. Our sample sizes for our studies are usually constrained. It's not offered clinically. There's no kind of reimbursements or insurance for these assessments. It's really difficult to get real-time results. Once we go to all the effort of collecting all our samples, we send them for assay. It takes weeks to months to get the results back. So if you need to know that person's body clock time now, it's kind of too late. And we're limited in how often we can measure people's body clock time outside of these carefully controlled lab conditions. So we don't know a whole lot about day-to-day changes in people's body clock time in more real-world conditions. So my field in particular has hit this really big roadblock, right? We really need practical alternatives to be able to track people's body clock day to day as they go about their normal life. So since my PhD and through my postdoc and still now, I've been working a lot with alternative approaches, particularly looking at different model-based tracking, which will use wearable data that you can collect from like accelerometers, heart rate, light sensors. And basically what they'll do is they'll take all of that data, all that time series data, and they'll put it into a model of some type, and they will generate daily predictions of circadian time. Because there are a lot of approaches being developed to do this, and there's a lot of interest in it, it's a big roadblock in our field, I've developed a little bit of a list for what I think the model needs to be able to do to be useful, because some are quite good and some are less good. So it needs to be simple and non-invasive for the participant. It needs to use passive sensors. It has to be easy to wear. It needs to be able to predict circadian time changes over time in real time, like it needs to be a dynamic model. And it needs to be accurate at the person level. A lot of them are quite good at a group average, but that's not particularly helpful if it's not accurate for the person that's sat in front of you. And then really importantly, it needs to be well validated in a really wide range of settings against reliable phase markers like melatonin so that we know that it's accurate. It's really important we do this in lots of cases. Not doing it is kind of like letting ChatGPT write your code and then not checking that it actually works. So we need to validate it broadly. So currently our best validated approach, in my opinion, is a dynamic systems model approach. It basically uses a series of differential equations to solve for phase over time. And essentially what it does is it will model the circadian pacemaker as an oscillator, and it will just continue to oscillate with a fixed period in the absence of any time cues, exactly like what our body clock will do. And then because light is the key synchronizer to the clock, we can put light into the model to produce a phase change. So our oscillations can be modified. We can push them longer to push the clock later, or we can make it spin faster and produce an advance, depending on the timing and the strength of the light stimulus that you give it. So this is a physiology-based model. It's grounded in how we know the physiology works, and it's dynamic. It responds to changes in the environment. So this model was developed and validated in the 90s using really, really carefully controlled data in really, really carefully controlled experiments so that we could learn how that light would affect the clock. And that's a really, really controlled test case. But what I like about this model is that it's now been tested in a lot of more challenging real-world test cases. So with data collected in real people out in the world, either sleeping at the same time, sleeping at all different times, some limited testing in clinical populations with some delayed sleep-wake phase disorder, and in shift workers who have lots of frequent disruption all of the time. And basically every time we see the same thing. We see that it is accurate within about an hour at the group average level, which is pretty good, it's not bad. But consistently, it doesn't have good individual person level accuracy. The R-squareds when you plot what the model gives you versus what you measured are not very good. So that presents an opportunity. And a factor that I don't think this has incorporated yet or considered yet that I think should be is circadian light sensitivity. So we know now that there's this huge variation between people in how sensitive their circadian clock is to light. So for example, in this study from 2019, they showed over 50-fold variation in light sensitivity in healthy young adults. And basically what that means is that your effective dose of light is altered. So for this person who's really highly sensitive, they might be sat in this room here and it might be saying like, go, 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 lights on, highly sensitive to this. Whereas you, if you're less sensitive, you might have exactly the same room lighting and it's barely registering to your circadian clock. And so we know that there's that variation related to different medications, different clinical groups with age. So there's all sorts of people out there getting effectively the same light patterns, but totally different doses of light to their circadian system. So that's something I'm super interested in exploring. It's basically looking at whether these differences in light sensitivity and how they interact with people's real-world light patterns can explain the individual variability we see in people's body clock time, and then can that be used to improve our person-level accuracy of our models that predict circadian timing. So I've got two quick little studies to show you. This is where the data trait comes in. So to look at this, what I do generally is we'll take our standard model of the circadian clock, it takes inputs of light data, and it will predict circadian timing each day. And then within that model, there are some parameters that alter the light sensitivity for the model. So we can change the shape of the dose response curve, so how sensitive the model is to lower levels of light. And we can change the shape of the phase response curve as well, so I can make it more sensitive to advancing or delaying light. So for the first study on this, we used data that we collected in people who lived on a fairly standard diurnal schedule, so sleeping at night time. We monitored their light exposure for 3 weeks using a wrist ActiWatch that had a light sensor on it. And we used that as an input into our model, which would predict our circadian timing. And then within that model, I played with our light sensitivity parameters, basically to see if we could find the right fit for each person. And then we compared the accuracy of the model to measured circadian timing. So we looked at dim light melatonin onset, so the rise of that melatonin profile at the end of each week. And when we run the model using our default parameters, which are all based on like the average person's physiology, this is what we get. So I've got our measured body clock time here and the predicted body clock time here. And it's doing okay. The mean absolute error is about an hour, pretty standard for this kind of model, but the R squared is only 0.2. So it's not doing very well at the individual person level here. But when we optimise our light sensitivity parameters in the model to find the best fit for each person, we get really large improvement, both in our average error, we're below half an hour now, but importantly in that individual level accuracy. We've got 78% of that variance now explained. At the person level, it's way more accurate. So to us, this is evidence that we can account, by accounting for differences in light sensitivity combined with what people's real light behaviour is, it explains a large amount of that individual variability and where their body clock is sitting. And it also suggests that if we can find a way to measure someone's light sensitivity, that's a really promising way to find a good person level parameter set for them to be able to track their circadian timing long term. So now the second study on this, we were looking to see how this goes in shift workers, which you might remember, they experience a lot of circadian disruption. They are therefore a much trickier test case for the models because there's way more noise in the signals going in. They're also a population where it would be really useful to be able to track their body clock time for various countermeasures. So we got an ARC grant and collected all this data during my postdoc with Andrew Phillips at Monash. The data collection we wrapped up last year, and we're kind of busily analysing lots of data at the moment. But the basic protocol was we monitored shift workers for 4 to 6 weeks. First, when they were on a day active schedule, which we called our baseline, and then on their usual shift work roster, which was anything from nurses working rotating day, evening, night shifts. We had police officers who worked 7 night shifts in a row, which was horrifying. We had paramedics who will do 2 days, 2 nights, 4 off rosters. We had all sorts of different shift work rosters in there. And then over that time, we were measuring their sleep timing and, importantly, their light exposure all hours of the day using wearable devices. And then we also had some pupil-based and some melatonin-based markers of their light sensitivity as well. And then, of course, we measured their body clock time 3 times as well in the laboratory, looking at their melatonin onset. I will note during the shift work portions, we had them in for full 24-hour constant routines, which means being awake and in the dark and in a bed, spinning in a tube every hour. And the reason that we have to do that with shift workers is their body clock could feasibly be anywhere in that 24-hour period, so we had to measure the full 24 hours to make sure we got it. So as you can imagine, this was quite a gruelling study. We got 18 participants completed, but we have really high resolution physiological data, particularly during those constant routines, and then really good time series data as well, which is very exciting for me playing with our phase prediction models. So this might be a bit tricky to see, but bear with me. I'm showing you 4 raster plots of 4 different shift workers who went through this protocol. Each row is about a day. The grey is when they're sleeping. The green is when they're working, so you can see this person went from sleeping at night to working at night. The little squiggles is their light exposure, minute to minute, and then the diamonds are their body clock timing. That's when their melatonin started to rise. And what you see is we've got this characteristic variability in circadian timing across these people. Like this person kind of didn't shift much even though they worked night shifts. This person shifted a little bit after some night shifts and then a lot after some other night shifts. These people just floated around all over the place. And while this is maybe hard to see, the main reason I want to show you this is I want to show you what the model does when I use each person's light data to predict their biological timing. So this blue line is now the predicted circadian timing each day from the model based on the group average, just default parameters. It's doing fairly well at baseline. It's getting everyone fairly spot on, but it really starts to lose the thread as we go across shift work. Some of these people whose body clock was delaying, it's not predicting it at all based on their light. Some of them it's kind of doing okay, but they didn't really shift much. So taking their light exposure alone wasn't enough to predict what was going on. But now in this pink line, what I'm showing you is when we adjust the light sensitivity levels in the model for each person. And what we found is we can really track the changes across this roster much more accurately for each individual person with their own individual sensitivity set. So this is more to show you at a conceptual level what we're hoping to do. We want a model that can track those daily changes in timing in a way that's accurate for each individual person, even when there's circadian chaos, there's all this misalignment going on. And we've got more data than this. I'm currently running the model over our other participants to see if we can start to optimise the model more based on our physiological markers of light sensitivity so that we can more prospectively go, this is the parameter set for you going forward. Cool. So in general, what we're seeing is differences in light sensitivity combined with our real-world light patterns can explain a lot of the circadian variability that we see in the wild, particularly in shift workers, and it can really dramatically improve how accurate our model is at the person level. We're doing a lot of work now really focusing on prospectively individualising those parameters, so like which parameter set do I need for you, looking at different pupil markers and different other markers that we can get about the person. And we're really working to validate it really broadly. Like I need this to be validated in wide data sets, people who sleep at all different times. I want as much circadian chaos as possible to throw at it so that we can really be sure that it's working and it's going to generalise. And I think once we've done that, it's going to be a really useful tool in a lot of cases. Some of these are shift work based, like personalised chronotherapies for our shift workers across different sleep disorders, but also potentially across a lot of other cases that I haven't listed here where body clock timing is relevant. We can couple them with models of our sleep and alertness as well. So from a fatigue risk management point of view, we can have much better evaluation of what work rosters are safe, what's not, when do people need countermeasures. And also just in general to support healthy light diets, as we're starting to call them, for better sleep and circadian alignment in general. So we're looking to collect loads of data. I just want to acknowledge various mentors and collaborators and, of course, the John Trinder Sleep Lab here for welcoming me. Thank you.
Thank you, Julia. That was really fascinating. We can take maybe 2 questions while we transition over to Olivia. Martha?
Yeah, really great talk. I understood that you used the light sensitivity to improve the model parameters, but it wasn't clear to me how you measured that for each individual, and also how did that optimization fit in the model for each individual?
It's a really good question. That's kind of what we're working on at the moment. So that preliminary one in the shift workers is me going, I think maybe you're this parameter. Like I've not measured something and gone, therefore it's this. But we have got measures of pupil-based sensitivity and some melatonin suppression things that we're starting to develop functions that basically will map like, okay, if we have this physiological range in this type of sensitivity, does this map onto this parameter well, and then testing it that way. That's kind of what I'm working on at the moment. It's kind of the bigger challenge is getting enough data to be able to do that with. The first one was much more like a least mean squares, which one gives the best fit for each person as well. So not prospective. Yeah.
Thank you very much. Very interesting talk. I have more of a methodological question. So sitting 24 hours waiting to measure melatonin sounds like torture.
Yeah.
Is it possible to just like light wax the phase out? Can you just give somebody a lot of melatonin, which is artificial drug, to whack the system so that you can maybe calculate and model the change from the normal level? So is it something that is possible to experimentally shorten this process?
I don't think so, because we're trying to measure a marker of the internal clock rather than melatonin itself. So if we give you a lot of melatonin, we will see lots of melatonin in the assay, but it's because I've given you a lot of melatonin, not necessarily that that's where your central clock is sitting, if that makes sense. We can measure melatonin usually with as little as 7 hours, which is kind of what we're doing at the moment, which sounds way better than 24. But yeah, they're pretty grueling studies to run, and so that's kind of why everyone's looking for other options. There's a bunch of metabolomic approaches as well that people are looking at to look at, if I take 1 or 2 24-hour matched blood samples, do the profiles in the different RNA clock genes give you a sense of timing as well? So like, there's hope that we could do it with much fewer samples like that as well. Harder to induce experimentally though. Yeah.
Thank you so much. Please stick around and ask more questions. All right, so now we're going to hear from Professor Olivia Carter. Olivia is going to talk to us about something a bit different today. She's going to introduce the Melbourne PX4 Health Initiative, and I'm really excited to hear about this. It has the ambition of establishing a world-leading integrated medical research capability to advance personalized medicine and well-being at scale. Olivia has held NHMRC and ARC fellowships, and she has conducted research in neurobiological and psychological factors that influence the conscious experience. But today she's not going to talk about that work. She's going to talk about this initiative, which we're all really excited to hear about. So thank you, Olivia.
Thank you. Thank you for having me, and thank you for turning up. So yes, and I might just put a placeholder that we're actually sort of doing a bit of a branding exercise, because the PX4 Health, there's a lot of acronyms in all of this, OMIX3 and PX4 and different things, but at the moment it's the PX4 Health Initiative. So I'll start by giving a little bit of a whirlwind background to the problem that the PX4 Health Initiative is aiming to address. Now, this is actually a slide that was given to me by a data science friend. I was actually surprised to discover this through this process. I've been learning a lot, learning about multiomics that I never thought I'd be exposed to. But it's really quite extraordinary if you stop to think about this, that we start off as a single human cell. As an adult, we end up having around 30 trillion human cells, but the adult human body includes around 100 trillion microbial cells. So about 70% of you is not you. So just thinking about that in terms of understanding the complexity of the factors that impact your biological health. I suspect it's not really news to anyone. This is not a controversial claim at all, that there's a lot of factors that are clearly being, there's clear evidence linking a lot of factors like exercise, environmental exposure, socioeconomic stress, and available resources, access to medical care, diet, pathogens. All of these things are known undisputedly to impact health. The problem is really understanding what's the relative contribution of these things, particularly when it comes to things like chronic disease. There's normally multiple different factors that are making things worse or better, and really trying to understand in terms of health, optimising health, disease prevention and other things, really to understand which are the biggest determinants or the biggest factors, and in general, often these things will change across the lifetime as well. So these sort of understanding the relative contribution of each of these different factors in these complex sort of real life scenarios is extremely difficult, but we are now at place where that these sorts of problems can start to be unpacked using the power of big data and AI. But it says down there in very small text that the capability, really AI at the moment, if you just grabbed all of the data from all of the studies that have been done across this Melbourne precinct or across the world, the AI models would run, they'd find differences across the different populations, and they'd be largely trivial, non-interesting differences that the cancer cohort, that all their measures were done on this sort of equipment or stored in that sort of way. So it's been literally trillions of dollars have been invested globally into trying to use current AI tools to interrogate existing large data sets, and it really doesn't work because of the noise in the data. So the question is, if we know that there's a lot of complexity and many factors impacting human health, disease and longevity, how can we use technology to improve quality and efficiency of data collection, integration, analysis to unpack complex problems? So that's really I guess the backdrop to the PX4 health initiative. And I'll try to zip through this relatively quickly and then spend a few slides on each of these different elements. So the way we're sort of conceptualising this and addressing this is to basically establish some dedicated testing facilities and I guess data generation facilities. And the moment, as I said, we're looking into more friendly names, but the term platform is important. Yeah, deep phenotyping platform and other things is not so intuitive, but it does distinguish between this sort of core research infrastructure, which is often labelled sort of platforms in the university world, and accesses different sorts of funding, national funding. So that's really the idea, is to establish the core capabilities that will be of value to academics across the university and wider community. All right, so I will delve into each of these things. Actually, no, I don't really talk too much about this bottom thing, so I'll just add it here, that once you've collected a huge amount of data and you're bringing people in and they're giving up their own time to come in, it's a really important element of this whole exercise, is to ensure that useful information is being provided back to participants. So we haven't really fleshed this part out, this bottom part out too much yet, because we're still sort of the very early stages of data collection, but just to say that that is something that's well and truly on the radar of thinking about ways to provide information back to participants, but also to use this as a research opportunity where we have people coming in to actually start understanding, asking them to what sorts of information they would like to know about their health. If we're getting to the point where we can analyse biological samples and learn about their lipids and metabolites and all of these things, do people want to know those things? Do they understand that information? Is it overwhelming? Is it positive? Is it not? So there is an opportunity to actually really look at what is the most valuable way to provide information back to make real sort of health changes. So just a quick slide, that a major goal of this whole initiative is to integrate the local expertise and capability across the entire precinct. So this is just our campus. We're over here, as you'll see. A lot of this has involved MCRI, down the children's, but I just on this slide to flag law. There's a lot of people ask. It's an extremely important question around medical data security and privacy, and really thinking about those things. And so we've had a great support from some of the legal experts, professors in law that specialise in those areas. So it's really, the idea is to say there's a huge amount of local expertise here, and if we can really, I guess, bring that together and have these resources that are then available to everybody else, that are set up in a way that we know are validated and are at the highest levels of sort of data security and other things, then it'll be hugely valuable. So I'll go into the data in a minute, but just to say that the goal is to set up these this sort of research infrastructure, basically, and then to establish a large community sort of representative sample. We've got the non-clinical sample, but nobody is perfectly healthy, so it's more the sort of community representative sample, but ideally also be supporting the assessments of other clinical cohorts that are currently being tested. Basically, you know, every individual research group sets up its own trials, facilities, and reinvents the wheel over and over. So that will be the goal. And then if all of these different cohorts and the community sample have data collected effectively on the same way and prepared in the same way, then the participants can be directly compared against each other across different cohorts. And that itself saves a huge amount of time and resources in terms of trying to identify appropriate controls for any clinical cohort coming through. And just to say at the bottom that the goal is also to align as much as possible with some of the really large international biobanks like the UK Biobank, which many people have probably heard about. Okay, so these are the four sort of key elements. As I said, I'll dig into them, but the first is this sort of dedicated clinic facility. We need a place to store a lot of the samples, the biosamples, which at the moment we're working with MCRI, has this huge, it's really huge, it's about half as big as this room, and a multi-omics platform. I'll go into what is multi-omics in a second. Okay, and then onto the data storage. So we're now making a lot of progress. Here's a photo of Julian Simmons, who many of you will know, and he's probably currently over in Cardigan Street doing a lot of work here. So these are some older photos, but this facility is now totally sort of kitted out, as it were, and we've had the first 150 people coming through in a sort of pilot testing. There's still around 4 hours people are coming in. That's really, at the moment, mainly collecting biosamples and doing a reduced scope of assessments. I'll talk a bit at the end about the next phase. We're now at the point of, I guess, reconsidering all of the different measures that have gone in. At the moment, there's no cognition. There's a lot of the things that we would consider relevant to psych are not currently in there, and that's largely because, well, first of all, the funding, which I'll get to, was there's a lot of funding going into this multi-omics and the biosample collection. And at the moment, we're bringing people in for 4 hours, and it's really managing the blood collection, getting samples over to MCRI quickly enough. So now that a lot of that has all been smoothed out, we'll introduce, we're sort of going into the next phase of a full scope of the phenotyping assessment. Okay, so for those of you who don't know, this is physically at the Australian College of Optometry, which is sort of over, it's a block or so away from psych, so it's relatively close. And just in terms of the sort of work that's being done at the moment, there's a huge amount of work just setting up the infrastructure. We've got a data manager. Again, this is work that Julian's really leading. In terms of pulling in, there's Fitbit wearable data, health apps, setting up REDCap, participant management systems, all of these things, so that in the next year or so we'll be able to just support if other groups say we've got running a trial or we're interested to collect X or Y data, we've got 50 people, we should just be able to sort of slot them in, have everything set up. Okay, the multi-omics, I've got a video about what is multi-omics, but just to say, I guess the multi-omics platform is really the centerpiece in that it's something that on the global scale, Melbourne University and Melbourne as a city is really world leading. So we were lucky enough to get a $15 million grant, sort of 30 million in total when you bring in all the university and other in-kind. So that was $7 million from the MRFF, but included funding from industry and university. In 2023, just to say that the School of Psychology put in $600,000 to really kickstarting all of this together with the School of Biomedical Sciences, and that money was actually helped, we sort of leveraged that money in this grant application, and also just to actually start getting things off the ground in terms of the clinic facility. So that the grant funding was received mainly to purchase this very fancy equipment and to fund staffing around the use of that equipment. But I do need to acknowledge there's been really impressive levels of support coming in from faculty at the moment. I'm not, we'll have to see. It's only ever 1 year at a time in terms of budgets, but this year that faculty's provided around $1.5 million if you include direct funds and also large equipment grant. I think we're recently successful with a DEXA, which is like a bone density and body composition scanning, which is useful to understand, sort of whole body health. Okay, so the reason we got the money, the $15 million, was, this is news to me, in terms of the major obstacle in terms of multi-omics analysis is that basically people get their samples and they'll say, okay, I want to understand the lipids. They'll send off some bloods over here to one provider and proteomics somewhere over there. And there's lots of different providers of equipment, and every time everyone runs any sort of analysis, they optimise the parameters and they change everything and twiddle everything. I'm now really sort of oversimplifying things, but we effectively got the funds to buy dedicated, the absolute cutting-edge facility equipment, and then optimise them for sort of efficiency, just high throughput analysis, but then set those parameters. And in setting those parameters, it means this will be the first sort of facility that will be possible to have accredited for clinical use. The clinical accreditation just requires standard operating procedures being used over and over and then validated over and over. So that was really, the grant is over a 3 to 4 year period to establish this pipeline and set it up as a clinically accredited facility, which also means that it will be possible to compare clinical and healthy cohorts as well as in the same individual across time. It's really that stability of assessment. And the other thing that is first in the world as far as I'm aware, there's I think a few groups trying to do similar things globally, is that a single sample, so single blood sample, or at the moment we're collecting urine, stool, saliva, plaque. There's been discussions about sperm, which I'm not sure is going to cut the mustard, but at the moment we're doing those things. And there was the other discussion was around tears, which they're collecting in the School of Optometry. All of those things are theoretically possible, including spinal fluid, but at the moment there's the standard plasma serum, urine, stool, saliva, plaque, and that single sample is then basically fractionated into the 3 separate tubes, but it's the same sample. They go off onto these 3 different systems and then have to be stitched together again. The integration of that data is a non-trivial problem. We're working together with the Australian Biocommons, which is it's effectively the national funding mechanism rather than a group of people. It funds a large sort of network of people that are really building out the data infrastructure to support genomic and other health-related data. So that team got a $25 million grant to do some work in that space, and for the government. And this project is sort of a flagship within that project, if that makes sense. So we're working really closely with them, and that's extremely helpful. Now, this is one reason I'm trying to stress this sort of every different group that's involved, because that's really the entire point, is that really trying to bring together these groups that are doing the kind of national scale types of contributions. Now, this is maybe not national scale, but one thing we said in terms of the grant is we were going to build this multi-omics infrastructure, but at the end of it, we're also going to be talking with the legal people and community user groups to make sure that at the end of this, we have communication strategies so that we can communicate what it is that multi-omics is. If in 4 or 5 years' time people are going to GPs and getting multi-omics assessments and they have no idea what multi-omics is, or they're terrified of it, that will be unhelpful. So we actually have teamed up with the third-year animation students from the VCA, and we were their test case. They were there as sort of pretend clients, but they've done a fantastic job. So we hosted them. This is the Bio21 lab where the multi-omics equipment is, and we showed them all the stuff, and then we worked with them over this first term, and they've created this video, and I was very impressed. We'll be working with them again the next crop next year. Okay, so this is a global premiere, I should say. So strap in, it's 2 minutes. Hopefully you know more about multi-omics at the end of all of this. All right, hang on. Is this loud enough? Can you hear it? It's probably...
Our body contains thousands of molecules that reflect our genetic makeup, our diet, and our lifestyle. Most blood tests that a doctor might order typically detect just one or a few of these molecules, such as glucose or cholesterol. But what if we could develop blood tests that measured many hundreds or thousands of these molecules at the same time, providing a better understanding of our state of health? The ability to do this on a small blood or tissue sample is fast becoming a reality, with new developments in multi-omics technologies. While technologies for sequencing our genomes to understand whether we are more likely to develop specific diseases are well established, changes in the levels of these molecules in the blood or in other tissues can be an early indicator of disease, while also providing a lot of new information into their underlying causes. Multi-omics can be used to diagnose diseases with greater confidence than traditional pathology tests, as well as to allow us to more precisely tailor treatments for each person. Results will give people the information they need to make lifestyle choices and help identify targeted medications that work best for them. Thanks to advances in multi-omics analysis, the future of medicine is becoming more accurate, more efficient, and more personal.
All right. Anyway, that was that. I was impressed of this third year students. Okay. Oh, we won't do that again. All right, I might skip through all of this for time and just say, I'll come to this in a second, but I did just want to stress, because this is a psychology audience, is that, well, first of all, there has been a focus on the sort of multi-omics element, but really where this starts getting exciting and interesting is to be able to really bring in the expertise of people in behavioural sciences and others to start looking at, we know that stress and mental health and all sorts of other things, lifestyle, impacts the physical elements within the body, but understand bidirectional relationships, but really trying to actually understand that and in what ways and how, the effect of age and other things will really be the power of this. So just to really quickly, just to make the point that what this multi-omics thing that we're establishing is really leveraging support from an entity called Bioplatforms Australia, which many people have not heard of, and it's the NCRIS, the National Critical Research Infrastructure scheme, and it funds things like synchrotrons and other massive scale infrastructure, and so we've been lucky to be supported by the BPA. They were a sort of co-funder in the grant we got, and they also, the Australian Biocommons is part of that group. And the thing that's really been to be really positive about all of this is that the BPA network, it's really about building infrastructure that's a value to the country, and that everything that is created and established is used as much as possible open source software and other things, so that's why they're helping us, is that the solving, the problems we have will be of value to other research groups as well. And just to say, so this is, BPA is a national sort of network, but with node leads, and the node leads sit within universities. It's just university academics, but they basically get this sort of direct funnel through from national funding if they can demonstrate that they're building things that are of value and being used by the research community nationally. And just to highlight, so one reason Melbourne is a good place to be, if you're interested in these things, is Melbourne has the node leads for the sort of genomics, proteomics, metabolomics and bioinformatics, and it's Malcolm, who's the head of Metabolomics Australia, is really leading the multi-omics component, and Bernie, who's the bioinformatics head of the Australian Biocommons, is leading the data science side. It's just one slide from that. So it's still very early days, but this is the kind of front page snapshot of the access that there will be to the data at the end of the day. So that's currently being built out at the moment. Finally, okay, I'm nearly finished. I did just want to say something about the Gen V cohort. So the idea here is that we'll have these platforms that I've talked about that will support sort of assessment of the individual clinical cohorts. And here I've just said that the goal is to also work with Gen V and try to recruit 10,000 of the parents of the Gen V cohort. And they, for those that don't know about Gen V, so it was a 2-year birth cohort that finished about a year ago, so that all the babies are about 1 to 3, and the parents are any age group that would have children between the age of 1 and 3. But they've got a lot of data and a lot of different, been recruiting with lots of different languages and other things. So it's a really be a fantastic opportunity to really extend that collaboration and try to get some rich data from the parents and the children. This is just a snapshot of where we're up to. So and I'm going to say it really is a we, huge numbers of people, I've got a slide in a second with all the people involved so far, has largely been this sort of operational stuff, getting facilities up and going, and a lot. I've been on so many Zoom calls about exactly which type of plastic tube should be the right plastic tube, and I'm sure there's much more complications about the exact specifications of the tubes. But anyway, there's a lot of discussions about those sorts of things, and this sort of sandbox, we called it the sandbox pilot. So 150 people have now gone through. That data will now be sort of worked on and analysed, and a lot of it is to look at sort of, data quality and other things. That data won't be released to the world community. It'll really be around building the right data infrastructure and running a few smaller analyses. The goal is that by 2026, this entire pipeline is now up and going in terms of the equipment. The second half of this year, we'll be running all the samples that have now been collected and are sitting in the biobank. And the goal is to bring through around sort of 500 or so healthy, community sample, not healthy, but then really by 2027 or so to be well and truly set up to be supporting recruitment and testing of other cohorts or other groups. So we are trying to really socialise that idea around, sort of writing grants and other things. If this is of interest, we should be in a place that we'd be able to, manage additional cohorts by then, and still a bit funding dependent, but that's where we're working towards. Just to say, we are, we've run this sort of reduced version of the phenotyping. We're now going back and reviewing all of the measures, and we'll add more with this complex sort of matrix of big issue is participant burden and duration. we just can't be doing 25-minute measures of everything at the moment. It's taking a huge amount of time, and also making sure that at the end of it we collect some data, that someone's actually interested in the data collected. So the sort of second half of this year will be a lot of that. This is just a list of the people that are currently directly involved in all the different things. There's a lot more people indirectly involved, and this list will grow. And that's it. That's it. So thank you.
Thank you, Olivia. We've had 2 talks talking about an extensive measurement within people, which is interesting, and different approaches. Does anyone have a question for Olivia? All right.
Thank you very much. Very exciting opportunity to study the body. But what about the brain? Is there any UK Biobank is known for its brain one? Any plans to extend this to an MRI session here and there, or perhaps an EEG resting state, 10 minutes, something like this, to just add the brain to the picture?
Okay, it's an excellent question. So we're in the process. So the EEG was not included in the initial thing. It's number 1 on my list of things to make a decision about, that we need to make a decision about, is whether there's any resting state EEG or at-home EEG overnight, and particularly in terms of sleep and other things. There's no MRI that's going to be part of the standard protocol, and one reason for that is because the UK Biobank has actually done a very, very good job, and there's a huge number of studies coming through, and that's great. The other reason is because it's massive participant burden to come in, and discovery-based, an MRI-based marker for discovery is not super translatable into the community. So there is a real prioritisation. First of all, exploiting the thing that Melbourne is globally sort of leading in around the multiomics, but also any assessment, if we find important markers like mentioned the chronotype, or if it's the blood-based marker, saliva-based marker. So there'll be a lot of assessments comparing not even, what a lot of the dental folk think you can diagnose cardiovascular disease based on, saliva and plaque. So the more we can get towards really easily testable sort of measures, the better. So there would need to be a really clear value in EEG data. We wouldn't be able to do the full kit and caboodle EEG, and to what extent there's value in a really reduced down version. If we're sending people home with a single-use EEG electrodes, it's not going to be easy to get them back. All this sort of stuff needs to be factored in. I will add, because I will forget this otherwise, that we actually met with Eli Lilly representatives when they came out, and one thing that was high on their list of things was pain, and was asking about sort of pain measures. But the truth is that a lot of the MRI stuff is very interesting, but it's not as clinically sort of useful. Yeah. Yeah.
We're happy to talk about it later.
Yeah.
Maybe I can. Another question?
Yeah.
The bad precedent, but today all up.
Where does this data become saturated? So there are lots and lots of omics and data points, but is there any time at which there's too much? So are there any principal components from this data that you expect to kind of, is there any data on this? So how does this large amount cluster into useful parcels, and can you reduce tests to those that give you the biggest output?
Okay, so everyone that has had any opinion about this, so I'm not an expert, but it's certainly the indication is the more people, the more participants, the better. Okay, the more human variability, ethnic variability, age variability, circumstance variability, the more you can also test the same person across time, the better. Also, everyone is sort of saying is that the expectation would be the more you can get information out of the lipids and the proteins of the more detail you can get out of blood sample, probably you're going to make the urine sample redundant, you know, and so that's a very first pass thing that will be done in the initial analysis. But the cost of storing huge volumes of digital data, if there's large numbers, I mean, it's in the millions of dollars per year to store the data, okay? So the data reduction in terms of the multiomics is a very high on everybody's agenda, and there are people in maths and stats, like that's what they do, is just reduce down large multiomics data sets. So that will be happening, but then to have the sort of interesting signals amongst those data sets for the most number of people, and then to be able to compare this data with UK Biobank and other things, it's just the more data, the more power.
Thank you so much for coming. We'll have another colloquium next month with Candace Blake and Sophie Lynn, so please have a look out for September 2nd. Thank you to Olivia and Julia for fascinating talks. Please grab